The emergence of the first members of the genus Homo, beginning with Homohabilis approximately 2.4 million years ago in East Africa, marked not only a cognitive and technological turning point but also a biological gamble. These early humans faced an environment that was relentlessly hostile to fragile bodies unprotected by modern medical interventions. Infection, injury, malnutrition, and exposure were constants. Fossil evidence reveals the precariousness of survival. Skeletal remains often show signs of trauma, poorly healed fractures, or pathological lesions caused by chronic infection. Yet, despite such pressures, these hominins endured long enough to establish a lineage that would expand across continents. The risks they faced were immediate and unforgiving. Simple lacerations from animal attacks or falls could lead to septicemia in the absence of antiseptics. Gastrointestinal infections, now readily prevented with sanitation and antibiotics, would have been common in groups living close to water sources contaminated by animal waste. Dental evidence from early Homo erectus specimens demonstrates extensive wear, abscesses, and tooth loss, conditions that would have caused chronic pain and hindered nutrition. The lack of specialized tools in the earliest phases of human evolution compounded these vulnerabilities. Before the refinement of stone flakes into reliable cutting or scraping instruments, treatment of wounds was rudimentary at best, consisting of little more than natural clotting and exposure to the elements. High mortality characterized these populations, yet persistence suggests a form of resilience built not on medical knowledge but on adaptation and selective survival. Paleoanthropologists analyzing remains from sites such as Dmanisi in Georgia, dated to 1.8 million years ago, have noted individuals who lived for years despite profound physical impairments, including tooth loss and degenerative disease. Such findings imply that small social groups were capable of sustaining members who could no longer forage or hunt effectively on their own. The absence of medicine did not equate to the absence of care, though the methods were improvised and without formal understanding of pathology. Parasitic infections almost certainly shaped the biology of early humans. Soil-transmitted helminths, waterborne protozoa such as Giardia, and blood parasites like Plasmodium were endemic in tropical regions where hominins evolved. Genomic studies of modern populations reveal traces of these ancient pressures. For instance, polymorphisms associated with resistance to malaria predate the agricultural revolution, indicating that proto-human groups already bore the burden of vector-borne disease. The immune system of these early populations was therefore in constant training, repeatedly exposed to pathogens in ways that both culled the weak and fortified the genetic legacy of survivors. The skeletal record further testifies to the ubiquity of trauma. Healed fractures of long bones, as identified in Homo neanderthalensis and early Homo sapiens fossils, suggest survival through injuries that should have been incapacitating in the absence of splints, surgical intervention, or antiseptic dressing. Some researchers propose that natural clays with antimicrobial properties, known from African sites where early humans lived, may have been applied to wounds by chance. Such environmental resources could have reduced bacterial load, though without systematic knowledge of infection. The survival of individuals with serious head trauma, evidenced by cranial depressions that show signs of bone remodeling, demonstrates that even without deliberate surgery, the body's natural healing capacities occasionally sufficed when the individual avoided secondary infection. Nutritional vulnerability was another determinant of survival. Foraging diets were unpredictable, subject to the seasonal availability of fruits, tubers, and hunted game. Deficiencies in micronutrients such as iron, iodine, and vitamin D are thought to have been widespread. Archaeological isotopic analysis of bone collagen provides evidence for variable protein intake, with periods of scarcity leaving measurable biochemical signatures. Malnutrition compounded susceptibility to infection, creating feedback loops in which disease and starvation reinforced one another. Yet variability also forced versatility. Groups that could exploit a broad range of ecological niches, riverine fish, gathered shellfish, or marrow from scavenged bones, displayed higher resilience in unstable climates. This dietary plasticity became one of the foundational survival traits of the genus Homo. Beyond physiology, the organization of early groups affected outcomes. The estimated carrying capacity of bands of 20 to 40 individuals created both advantages and vulnerabilities. A small group could move quickly in response to ecological pressures, but epidemics or accidents could easily decimate numbers. Mortality rates among children were particularly severe. Paleodemographic reconstructions suggest that fewer than half of infants survived to reproductive age in early human populations, a figure consistent with ethnographic studies of hunter-gatherer societies without access to modern medicine. Such losses underscore the degree to which survival hinged on producing a surplus of offspring, ensuring that at least some would endure to maturity despite the omnipresence of death. These precarious beginnings framed the evolutionary stage upon which all subsequent medical practices, herbal remedies, surgical interventions, systems of belief, would later appear. The absence of formal tools of healing did not prevent survival, but instead sharpened the interplay between biology and environment. Those who lived long enough to reproduce were not necessarily the healthiest by modern standards, but were the beneficiaries of chance, resilience, and the minimal support structures afforded by kinship. Out of this crucible emerged the earliest patterns of human adaptation, laying the groundwork for the selective forces that would shape both body and behavior. The story of how humanity endured without medicine cannot begin with cures, because there were none. It begins with exposure, loss, and persistence against the odds. Each infection that failed to kill, each bone that healed in alignment by luck rather than knowledge, and each child that reached adulthood despite systemic vulnerabilities became part of a biological archive. That archive was written into genes, etched into skeletons, and encoded into the survival strategies of groups who faced a world with no buffer against disease or injury. The precarious balance of these early lives, sustained without intervention or systematic treatment, set the conditions under which immunity itself became the decisive arbiter of survival, shaping lineages through cycles of mortality that pressed relentlessly upon the species. The biological pressures that defined early human survival were never static. Pathogens evolved as relentlessly as their hosts, and the interplay between infection and resistance became one of the principal drivers of human variation. Immunity, in its most primitive form, did not arise from conscious intervention, but from the slow filtration of life through the sieve of mortality. Each generation that endured carried forward genetic configurations better suited to withstand certain microbial threats, while those who lacked such fortuitous traits left no descendants to propagate their vulnerability. Evidence for this dynamic lies embedded in the modern human genome. Regions of the DNA associated with immune responses, particularly within the major histocompatibility complex, MHC, display extraordinary polymorphism, reflecting a long history of selection by diverse pathogens. The extraordinary diversity of MHC alleles ensures that populations, rather than individuals, maintain resilience against epidemic disease. Molecular clock studies suggest that many of these variants were present in archaic hominins, and genomic comparisons indicate that interbreeding with Neanderthals and Denisovans further enriched the immune repertoire of Homo sapiens. The introgression of alleles, such as those influencing toll-like receptor pathways, provided tangible survival benefits in regions where exposure to novel pathogens was unavoidable. Selective episodes are visible in historical case studies. The persistence of the sickle cell trait across sub-Saharan Africa exemplifies the cost-benefit balance imposed by pathogens. Individuals heterozygous for the mutation in the HBB gene exhibit resistance to severe Plasmodium falciparum malaria, while homozygous carriers develop sickle cell disease with debilitating consequences. Population genetics has demonstrated that the high frequency of this trait is not an accident but the result of balanced polymorphism, in which the survival advantage conferred against malaria outweighed the costs imposed on a minority of individuals. Comparable dynamics are evident in the distribution of the Duffy antigen receptor for chemokines, DRC, where gene variants protect against Plasmodium vivax. Historical epidemics magnified these pressures. The plague pandemics that swept across Eurasia during the Middle Ages left imprints detectable even in present-day European genomes. A 2022 study of skeletal remains from London and Denmark, spanning the Black Death period, revealed significant allele frequency shifts in genes associated with immune regulation, including IRAP2. The observed changes occurred within a matter of generations, underscoring the intensity of selective forces exerted by pathogens of high lethality. Survivors did not merely endure. They transmitted altered genetic landscapes that recalibrated the immune system of subsequent populations. The immune mechanisms shaped by such episodes were multifaceted. Innate defenses, phagocytosis, complement activation, and the inflammatory response offered immediate though non-specific protection, while adaptive immunity introduced the capacity for long-term memory. The latter innovation distinguished vertebrates, and in humans it became one of the crucial factors in surviving recurrent exposures. Antibodies produced after initial infection provided rapid protection during subsequent encounters, allowing groups to weather outbreaks more effectively once an initial wave had subsided. Yet this advantage carried hidden risks. Pathogens such as influenza evolved high mutation rates to evade recognition, keeping the arms race in perpetual motion. Anthropological studies of isolated populations illustrate how fragile immunity could be when novel pathogens breached barriers of geography. The demographic collapse of indigenous populations in the Americas following contact with Europeans illustrates the vulnerability of immune systems unprimed by prior exposure. Smallpox, measles, and influenza, relatively survivable to those with ancestral experience of them, proved catastrophic to populations whose genomes had not been conditioned by earlier encounters. These events illuminate how deeply immunity is tied not only to individual health, but to the very survival of societies. Experimental science has confirmed the molecular basis of many of these historical patterns. In the mid-20th century, advances in immunohematology identified blood group antigens not only as transfusion determinants, but also as potential modulators of disease susceptibility. The ABO system, for instance, influences vulnerability to cholera and norovirus, with epidemiological studies from Bangladesh showing that individuals with type O blood experience more severe outcomes of Vibrio cholerae infection. Such associations underscore the subtlety with which pathogens have sculpted the human genome, often in ways invisible to daily experience yet decisive during times of epidemic. The very process of immune adaptation was not without collateral damage. Autoimmune disorders, in which the immune system mistakenly attacks host tissues, appear to be linked to genes that once offered heightened protection against infection. The hygiene hypothesis, advanced in the late 20th century by David Strachan, posits that reduced microbial exposure in modern industrialized societies has unmasked these liabilities, producing rising rates of asthma, allergies, and autoimmune disease. If correct, it would mean that traits once advantageous under pathogen-rich conditions become maladaptive when the microbial environment is attenuated. Population bottlenecks also shaped immune variation. Genetic evidence from mitochondrial DNA and Y chromosome studies suggests that Homo sapiens experienced severe reductions in population size during climatic upheavals, such as the Tobar supereruption approximately 74,000 years ago. Such contractions intensified the role of selection, as only a fraction of genetic diversity passed through these narrow gates. What survived was not necessarily optimal in absolute terms, but sufficient under prevailing ecological conditions, embedding the contingencies of history into the biological fabric of the species. The process of immunity, then, was not a guarantee of safety but a calculus of probabilities, operating across generations. The immune system hardened in places where exposure was relentless, and faltered in places where isolation offered temporary reprieve. Every epidemic, every endemic infection, and every chronic exposure nudged the human genome into configurations that carried both protections and liabilities. The record of these selections persists in the molecular architecture of the body, shaping not only survival against ancient diseases, but also responses to modern ones. Yet immunity did not operate in isolation. Human beings continued to encounter the natural environment with curiosity and necessity, compelled to identify external resources that might ease the burden of disease. Survival was not secured solely by inherited defenses. It was also influenced by what could be drawn from the plants, soils, and minerals surrounding early societies. The transition from passive genetic inheritance to active environmental experimentation became another critical dimension of endurance, intersecting biology with culture in ways that would prove decisive for the generations that followed. The human reliance on environmental resources for survival extended beyond food. In landscapes filled with roots, leaves, barks, and resins, plants became the earliest and most consistent external allies against illness. Their bioactive compounds were not identified through chemical analysis or laboratory testing, but through generations of observation, chance encounters, and often painful experimentation. Archaeobotanical evidence demonstrates that long before the emergence of agriculture, humans and their hominin relatives were already exploiting plants for therapeutic purposes. The first hints come from the fossil record. In 1998, a study of dental calculus from Neanderthal remains in Shanidar Cave, Iraq, revealed microscopic plant particles consistent with yarrow, Achillea millefolium, and chamomile, Matricaria species, both known for their anti-inflammatory and digestive properties. These were not common dietary plants, suggesting deliberate ingestion for medicinal effect. Additional analysis of Neanderthal teeth from El Sidron in Spain, published in 2012, identified chemical traces of poplar bark, a natural source of salicylic acid, the precursor to aspirin. The presence of these compounds implies not only awareness but selective use of plants with analgesic potential, centuries before their isolation in modern laboratories. Humans, through trial and error, gradually mapped the pharmacological landscape of their environment. The bitter taste of alkaloid-rich plants offered one avenue of discovery. Many toxic compounds, such as quinine from cinchona bark or morphine from the opium poppy, present as intensely bitter, and while high doses could be harmful, careful administration produced striking effects. The recognition that certain preparations alleviated fever, dulled pain, or reduced swelling laid the groundwork for a rudimentary pharmacopoeia. Knowledge of dosage was not standardized but transmitted orally, embedded in cultural traditions that preserved effective practices while discarding those that proved fatal. Chemical analyses of ancient residues provide further evidence of deliberate medicinal use. A 5,000-year-old Neolithic grave in Slovenia contained traces of Papaver somniferum, opium poppy, long recognized for its sedative qualities. In the highlands of Ethiopia, archaeological sites dating to early Holocene periods have yielded remains of Enceite ventricosum, a plant whose fermented starches were combined with leaves rich in tannins and saponins, compounds that exhibit antimicrobial activity. The distribution of these plants in burial contexts suggests that their use carried both medicinal and ritual significance, reinforcing the entanglement of biology with cultural frameworks. Willow bark represents one of the most striking examples of continuity between ancient practice and modern pharmacology. Hippocrates, writing in the 5th century BCE, recorded the use of willow leaves to reduce fever and pain. Centuries later, Edward Stone in 18th century England systematically studied powdered willow bark and reported its efficacy against ague, a term encompassing febrile illnesses such as malaria. This lineage of observation culminated in 1828, when Johann Buchner isolated salicin from willow bark, and eventually in 1897 when Felix Hoffmann at Bayer synthesized acetylsalicylic acid, producing aspirin. The arc from Paleolithic ingestion of bark to industrial synthesis illustrates the deep temporal continuity of botanical medicine. The functional mechanisms behind many of these early remedies are now understood at the molecular level. Garlic, Allium sativum, widely used in antiquity, contains allicin, a sulfur compound with potent antibacterial and antifungal properties. Turmeric, Curcuma longa, prominent in South Asian traditions, contains curcumin, whose anti-inflammatory effects have been demonstrated in modern pharmacological studies through inhibition of nuclear factor kappa B pathways. The persistence of these plants across disparate cultures suggests convergent recognition of their efficacy, shaped by direct experience of symptomatic relief. The adoption of psychoactive plants underscores another dimension of early pharmacology. The chewing of coca leaves in the Andes, documented archaeologically as early as 6,000 years ago, provided both nutritional supplementation of calcium and mild stimulant effects from alkaloids that alleviated fatigue and altitude sickness. Betel nut chewing in South and Southeast Asia, attested archaeologically to at least 10,000 years, provided psychoactive stimulation and antimicrobial benefits to the oral cavity. Such practices blurred the boundary between medicine and ritual, with physiological effects intertwined with cultural meanings that reinforced their use. Plant use was not limited to ingestion. Poultices, infusions, and fumigations represent early methods of external application. Honey, although not botanical, was frequently combined with herbs to dress wounds, capitalizing on its osmotic antibacterial properties. Resins such as myrrh and frankincense, harvested from trees in arid regions, were used to embalm bodies but also applied to inflamed tissues, their terpenoid compounds exerting antiseptic effects. Charcoal from burned plant material provided absorbent surfaces for treating gastrointestinal disorders, anticipating later uses of activated carbon in toxicology. The oral transmission of plant knowledge required cultural mechanisms for preservation. In small bands of foragers, the memory of which plants healed and which poisoned could mean the difference between survival and extinction. Ethnobotanical studies of contemporary hunter-gatherers, such as the Hadza of Tanzania or the San of southern Africa, illustrate the complexity of such knowledge systems, with hundreds of species identified for specific uses. It is likely that early human groups operated with similar sophistication, though their pharmacopoeias have largely been lost to time. The effectiveness of plant remedies was not uniform. The concentration of bioactive compounds varied by season, geography, and preparation method. Some plants required fermentation or heat to activate their properties, processes that could only have been discovered through long cycles of experimentation. Others carried lethal risks when misused, such as aconite, Aconitum napellus, whose alkaloids can cause cardiac arrest in small doses. Yet even these dangerous species were harnessed in controlled contexts, as aeropoisons or in ritualized medicine, demonstrating that early humans were not merely passive gatherers but active experimenters navigating a chemical landscape. The reliance on plants as sources of healing did not end with subsistence groups. As populations grew and settled, the role of flora expanded into organized traditions, preserved in texts, codified in rituals, and exchanged along trade routes. The first pharmacies of humanity were not institutions of stone but the surrounding ecosystems, transformed by knowledge into reservoirs of therapeutic possibility. And while plants alleviated pain, reduced fevers, and combated infection, humans also faced the persistent challenge of physical injury and the need to restore the integrity of broken bodies, a problem that demanded strategies distinct from chemical relief and directed attention toward the mechanics of healing itself. Human survival depended not only on resistance to disease, but also on the capacity to withstand trauma. Physical injury was unavoidable in a world shaped by hunting large animals, navigating uneven terrain, and defending against predators or rival groups. Bones fractured, skulls cracked, and soft tissues tore with regularity. Without intervention, many of these injuries would have led to death from infection, immobility, or hemorrhage. Yet archaeological and anthropological evidence indicates that early human groups developed techniques, rudimentary but effective, that enabled significant numbers of injured individuals to survive and return to functional life. Skeletal remains provide the clearest testimony. At the site of Shanidar Cave in northern Iraq, the skeleton of a Neanderthal male, dated to approximately 45,000 years ago, shows a crushed right arm that had healed in a shortened and deformed position, along with injuries to the skull and leg. Survival into adulthood with such impairments indicates both physiological healing and the likelihood of communal assistance. Similar examples of healed fractures, often of the femur, a bone whose break without support is typically lethal, have been documented in Homo sapiens remains from Pleistocene contexts across Africa, Europe, and Asia. These cases reveal that splinting and immobilization practices, however rudimentary, must have been employed to facilitate bone repair. The materials available for immobilization were dictated by the environment. Wooden sticks, reeds, strips of hide, or woven grasses could be bound around an injured limb to stabilize alignment. Clay, when applied in a wet state and allowed to harden, provided additional rigidity. Modern ethnographic studies among foraging societies show similar methods still in use, supporting the inference that early humans employed natural materials to replicate the essential principle of immobilization long before the concept was articulated in writing. Stability of the fracture site allowed the natural processes of osteogenesis, hematoma formation, callus development, and eventual remodeling to proceed without constant disruption. Wounds to soft tissue presented another persistent hazard. Before germ theory, the concept of infection was absent, yet experiential knowledge highlighted which substances aided recovery and which exacerbated harm. Honey, rich in glucose oxidase that generates hydrogen peroxide upon dilution, was a potent natural antiseptic applied to cuts and burns. Archaeological records from Egypt and Mesopotamia show honey listed as a common ingredient in early medical papyri. Beyond its antibacterial effect, honey's high osmotic pressure inhibited microbial growth while maintaining a moist environment conducive to tissue repair. Resins and plant exudates, such as pine pitch, were similarly adhesive, protective, and mildly antiseptic, forming natural dressings when applied to open wounds. Burn injuries, common in societies dependent on fire, elicited treatments that reveal considerable ingenuity. The application of clay and mud served both as a cooling agent and as a physical barrier against contamination. Ashes, containing alkaline compounds, were sometimes mixed with animal fats to create primitive salves, reducing pain and deterring insect activity around exposed tissue. These practices did not eradicate infection, but they improved the odds of survival by reducing bacterial colonization during the vulnerable early stages of healing. Trepanation, the deliberate removal of a portion of the cranial vault, represents one of the most striking and puzzling aspects of prehistoric wound care. Archaeological examples span continents, from Neolithic Europe to pre-Columbian Peru, suggesting that the practice was widespread and culturally embedded. Microscopic examination of trepanned skulls frequently reveals extensive bone remodeling at the edges of the openings, indicating survival for years after the procedure. While motives remain debated, ritual release of spirits, relief of intracranial pressure, or treatment of head trauma, the technical competence implied by survival rates is striking. Stone tools, when sharpened, were capable of cutting or scraping bone with precision sufficient to avoid fatal hemorrhage. The very repetition of this practice across thousands of years demonstrates that early humans not only improvised but institutionalized surgical techniques. Pain relief during treatment was limited but not absent. Alcoholic fermentations, derived from honey, fruits, or grains, produced ethanol that dulled sensation when consumed in sufficient quantities. Plant alkaloids with sedative properties, including those from opium poppy or mandrake, were occasionally available depending on geography. While inadequate for full analgesia, these substances could reduce distress, allowing procedures such as wound debridement or limb amputation to proceed with less resistance from the patient. The success of wound care often depended on the balance between promoting healing and preventing systemic infection. Chronic osteomyelitis, visible in skeletal records as pitting and abnormal bone growth, testifies to the limits of prehistoric treatment. Yet the very existence of healed fractures and surgically altered skulls underscores that survival was not an anomaly but a repeated outcome. Human biology, with its capacity for regeneration, was amplified by cultural practices that protected the body long enough for natural processes to complete their course. Evidence of amputation extends the picture further. A 2022 study of skeletal remains from Borneo described an individual who survived for 6 to 9 years after a surgical removal of the lower left leg, performed approximately 31,000 years ago. The clean cut and absence of fatal infection suggest not only technical skill but also postoperative care, including wound management and sustained provisioning. Such findings indicate that even in deep prehistory, humans possessed knowledge sufficient to perform complex procedures with a level of success previously thought impossible for societies lacking metal instruments or written medical theory. The broader implication is that early humans perceived injury not as an inevitable death sentence, but as a condition that could be intervened upon. Their methods lacked precision by modern standards, yet they reveal an experimental attitude. The repeated application of substances, the use of materials to stabilize, and the willingness to manipulate the body surgically. The persistence of these practices across continents and millennia demonstrates both continuity of need and convergence of solutions. While bones and wounds could be mended through physical interventions, illness often lacked visible cause. Internal fevers, wasting diseases, and unexplained seizures invited interpretations beyond the tangible, leading human communities to search for meaning in forces unseen. Where broken limbs could be splintered, invisible ailments demanded other forms of explanation and treatment, rooted in belief as much as in material practice. When physical injuries could be seen, touched, and manipulated, early humans devised practical measures to restore function. Yet much of human suffering resisted direct observation. Fevers, convulsions, wasting illnesses, and sudden deaths lacked visible wounds, producing conditions that could not be splintered or cleaned. In this gap between perception and pathology, explanations took shape that attributed disease to forces beyond the natural world. Spirits, curses, and unseen agents provided a framework for understanding, and with them arose the figure of the healer who mediated between the human and the invisible. The role of the shaman, though diverse across cultures, occupied a distinctive niche in the management of health. Archaeological findings from Upper Paleolithic burials, such as the elaborate interments at Dolní Věstonice in the Czech Republic, suggest individuals invested with ritual significance whose possessions included animal remains, pigments, and tools not primarily associated with subsistence. Anthropologists interpret these burials as evidence of early ritual specialists, individuals whose authority rested on perceived ability to communicate with non-material realms. Illness was often explained as the intrusion of hostile spirits or the loss of vital essence, and the shaman was tasked with restoring balance through ritualized acts. Ethnographic parallels among Siberian, Amazonian, and African societies illuminate the techniques employed. Rhythmic drumming, repetitive chanting, fasting, and the ingestion of psychoactive plants created altered states of consciousness interpreted as journeys to spiritual domains. The healer's trance was not merely performance but a controlled psychophysiological state, often accompanied by hyperventilation, sensory deprivation, or the use of substances such as Amanita muscaria mushrooms or ayahuasca brews. Neurobiological studies in the 20th century demonstrated that these practices induce measurable changes in brain activity, particularly in the limbic and prefrontal regions associated with perception and emotion, producing experiences of visions and dissociation that reinforced the healer's authority. The effectiveness of shamans cannot be evaluated solely by modern biomedical standards. Yet their practices often combine symbolic action with genuine therapeutic intervention. The administration of herbs, fumigation with antimicrobial resins, and the application of heat or cold frequently accompanied ritual. Placebo responses, physiological improvements driven by expectation and belief, likely amplified outcomes. Contemporary research has demonstrated that belief itself can trigger measurable changes in neurochemical pathways, including endorphin release and modulation of immune function. Within small groups where social cohesion was essential, The presence of a healer who inspired confidence may have improved survival rates by mobilizing both biological and psychological resources. Artifacts reveal that these roles were institutionalized across time and space. In the Americas, the use of tobacco in ritual healing is archaeologically attested more than 3,000 years ago, its smoke believed to purify and repel harmful forces. In sub-Saharan Africa, divination rituals often employed objects inscribed with symbolic markings, their interpretive use guiding decisions about treatment. Among Australian Aboriginal groups, healers known as Ngangkari employed both physical massage and ritual extraction of objects believed to embody illness. The continuity of these practices across widely separated populations underscores the universality of the need to make sense of unexplained suffering and to invest individuals with specialized authority to address it. The shaman's role extended beyond direct treatment of the sick. Rituals often encompassed entire communities, reinforcing shared belief systems and providing collective frameworks for coping with epidemic mortality or recurring hardship. Anthropologist Clifford Geertz described such rituals as models both of and for reality, simultaneously reflecting cultural understandings and prescribing responses. In this sense, shamanic healing not only sought to restore individuals, but also to stabilize the psychological fabric of the group, which was itself a determinant of survival in uncertain environments. Skeletal and artistic evidence adds depth to these interpretations. Cave art at sites such as Trois-Frères in France, dating to around 13,000 years ago, depicts anthropomorphic figures combining human and animal traits, interpreted by some scholars as representations of shamans in transformative states. The use of ochre, widely distributed in prehistoric burials, may have symbolized blood, vitality, or protection. These visual cues suggest that ritual healing practices were integrated into broader symbolic systems, inseparable from cosmology, identity, and social cohesion. Yet reliance on spiritual frameworks carried risks. Diseases caused by pathogens could spread unchecked when attributed solely to supernatural origin, and ineffective treatments might delay more practical interventions. At the same time, the persistence of shamanic practices across millennia indicates that the benefits, whether through genuine pharmacological substances, psychological reassurance, or enhanced group solidarity, outweighed the dangers. In environments where formal medical knowledge was absent, the fusion of ritual and remedy produced outcomes sufficient to allow continuity of populations. Modern clinical research has revisited these dynamics under the lens of psychoneuroimmunology. Studies in the late 20th and early 21st centuries demonstrated that social support, reduced stress, and belief in treatment correlate with improved immune responses and recovery times. These findings suggest that shamanic practices, though not based on empirical pathology, leveraged biological pathways that remain active in the human body. The distinction between symbolic and physiological healing is less absolute than once assumed. Ritual acts may have influenced survival in tangible ways. The shaman, then, was more than a figure of mysticism. He or she was a repository of ecological knowledge, an arbiter of symbolic meaning, and a practitioner of methods that straddled both the spiritual and the material. The persistence of this role across continents demonstrates its adaptive value in human societies confronting illness without formal medicine. Yet healing did not rely solely on specialists. The survival of the vulnerable often depended on the willingness of entire groups to share burdens, extending care beyond ritual into the daily practices of kinship and cooperation that sustained life. The survival of early humans depended not only on individual resilience or the interventions of ritual specialists, but also on the collective capacity of groups to sustain their members. Fossil and archaeological evidence reveals that individuals with severe impairments often lived long after injury or illness should have curtailed their existence, suggesting that caregiving was systematically integrated into human lifeways. This feature distinguished human societies from many other primates, where injured individuals are more often left behind. The Shanidar Neanderthal remains, particularly the skeleton of Shanidar I, illustrate this phenomenon with striking clarity. The individual, dated to approximately 50,000 years ago, exhibited multiple debilitating conditions: a withered arm, blindness in one eye, and significant hearing loss. Survival for decades with such limitations would have been impossible without consistent assistance in food acquisition, protection, and possibly even personal hygiene. Comparable cases of long-term survival with impairments have been documented in Homo sapiens fossils, including a 4,000-year-old skeleton from Vietnam displaying congenital spinal abnormalities, yet clear signs of extended adult life. Such findings point to an evolutionary advantage in cooperative care. Anthropologists studying extant hunter-gatherer societies provide complementary insights. Among the Ache of Paraguay, research conducted by Kim Hill and colleagues in the 1980s documented that elderly individuals, long past peak foraging ability, nonetheless received food contributions from younger members. The Hadza of Tanzania likewise engage in food sharing that ensures survival of children, the elderly, and the infirm. These ethnographic parallels suggest that cooperation in provisioning was a stable feature of human evolution, deeply rooted in kinship and reciprocity. Communal care extended beyond sustenance. Ethological studies of primates reveal rudimentary grooming and support behaviors, but humans elaborated these into structured caregiving roles. The presence of hearths, dating as far back as 790,000 years ago at Gesher Benot Yaakov in Israel, implies not only control of fire but also communal organization around warmth, cooking, and protection. Fire allowed injured or weakened individuals to remain integrated within the group's social space, rather than being abandoned in the wilderness. Neurobiological research sheds light on the mechanisms that underpinned this cooperative orientation. The evolution of oxytocinergic pathways, enhancing bonding and trust, created physiological incentives for caregiving behavior. Infants, whose survival required prolonged parental investment, reinforced the selection of nurturing tendencies that extended into broader kinship structures. Theories of inclusive fitness, first formalized by W. D. Hamilton in the 1960s, explain how genetic advantages accrue when individuals assist relatives who share a portion of their genome. Yet anthropological data suggest that care frequently extended beyond strict genetic kin, encompassing non-relatives integrated into bands through alliance or reciprocity. Burial practices further indicate communal attention to the vulnerable. The interment of children and disabled adults with grave goods, observed at sites such as Sunga in Russia, c. 34,000 years ago, demonstrates not only respect but also the allocation of group resources toward individuals unable to contribute materially. Such practices embody an awareness that survival was not reducible to immediate utility. The social fabric itself had value in sustaining continuity. Caregiving also influenced demographic resilience. High infant and child mortality, estimated from paleodemographic reconstructions to exceed 40% in many prehistoric populations, placed pressure on communities to distribute childcare responsibilities. Cooperative breeding models, advanced by anthropologists such as Sarah Hrdy, argue that the presence of grandmothers, aunts, and older siblings in childcare was essential for human reproductive success. By spreading responsibility beyond the biological mother, groups buffered against maternal death or incapacity, raising the likelihood of offspring survival in unpredictable environments. The psychological dimensions of communal care cannot be overlooked. Chronic pain, disability, or grief could undermine individual motivation, yet collective recognition of value, expressed through ritual, companionship, or shared food, maintained social integration. In small groups where every member was known, neglect was not easily concealed. The continuity of relationships generated accountability that translated into concrete acts of caregiving. Neuroanthropological studies suggest that reciprocal aid reduced stress responses, moderated cortisol levels, and indirectly bolstered immune function, linking social behavior to biological resilience. The archaeological record also hints at organized spaces for caregiving. Post holes and hearth arrangements at settlements such as Dolny Vestinus suggest designated domestic zones, which may have facilitated care for the sick and elderly. In later agricultural societies, written texts from Mesopotamia and Egypt describe households caring for the infirm, often guided by ritual prescriptions. While these texts reflect stratified societies, their underlying logic echoes far older traditions of collective support. The persistence of individuals with disabilities in the archaeological record is significant not merely as anecdote but as evidence of systemic survival strategies. A society capable of protecting those unable to fend for themselves gained advantages beyond compassion: the retention of knowledge. Elders, even if physically frail, carried memory of migration routes, seasonal cycles, and medicinal plants. Protecting them secured information critical for group adaptation. This pragmatic dimension reinforced the value of care, embedding it within both emotional and utilitarian frameworks. Yet communal care also imposed costs. Resources devoted to the infirm could strain groups already living at subsistence margins. Tensions between self-preservation and altruism likely arose, particularly during famines or migrations. Anthropological studies record instances of abandonment among some Arctic populations during extreme scarcity, suggesting that the ethic of care was not absolute but contingent on ecological pressures. The archaeological record may underrepresent such decisions, but the very survival of individuals with severe impairments indicates that more often than not, groups absorbed the burden rather than discarding it. This tension between cost and benefit shaped the trajectory of human evolution. Caregiving, while resource-intensive, enhanced group cohesion, stabilized demographic fluctuations, and preserved knowledge across generations. These dynamics created societies resilient not only through physical adaptation but through social architecture. Yet survival strategies extended beyond cooperative care, reaching into the sphere of diet and the deliberate use of food not only as nourishment but as a form of therapy, a recognition that what sustained life could also correct imbalance and restore health. The substances that sustained life were never neutral. They carried within them the potential to harm, heal, or transform. Food, more than a means of survival, became one of the earliest and most consistent instruments of medicine. Through patterns of observation across generations, humans came to recognize that diet could alleviate suffering, protect against illness, and strengthen resilience. This was not an abstract realization but a concrete outcome of lived experience, in which the difference between sickness and recovery often hinged on what was consumed. Fermentation stands as one of the most remarkable examples of food manipulation producing unintended medical benefits. Archaeological evidence from Jiahu in central China, dating to around 7000 BCE, reveals pottery vessels containing residues of fermented mixtures of rice, honey, and fruit. While initially serving as a means of preservation and intoxication, fermentation also generated lactic acid bacteria and yeasts that inhibited the growth of pathogens. Fermented foods such as yogurt, kefir, sauerkraut, and kimchi, known in later agrarian societies, carried probiotic organisms that colonized the gut, enhancing digestion and modulating immune responses. Modern microbiological studies have demonstrated that lactic acid bacteria produce bacteriocins and organic acids, lowering intestinal pH and restricting the proliferation of harmful microbes. The persistence of fermentation practices across continents underscores their dual role in nutrition and protection against infection. Spices provide another case where culinary practice intersected with antimicrobial efficacy. The wide distribution of turmeric in South Asia, cinnamon in the Middle East, and chili peppers in the Americas reflects not only preference for flavor but also biological effect. A landmark study by Paul Sherman and Jennifer Billing in the late 1990s analyzed traditional recipes from multiple cultures, showing that the prevalence of antimicrobial spices correlated with ambient temperature. In hotter climates where food spoiled rapidly, spices with strong antimicrobial properties were used in greater frequency, reducing the risk of foodborne illness. Turmeric contains curcumin, known to inhibit bacterial and fungal growth. Cinnamon produces cinnamaldehyde with broad-spectrum antimicrobial activity, and capsaicin in chili peppers disrupts microbial membranes. The culinary adoption of these substances demonstrates adaptive convergence between taste and survival. Beyond pathogen control, diet provided protection against nutritional deficiencies that carried profound health consequences. Skeletal remains from early agrarian societies often reveal lesions associated with scurvy, vitamin C deficiency, or rickets, vitamin D deficiency, illustrating the fragility of restricted diets. Yet in many cases, traditional practices evolved to counterbalance these risks. The Inuit diet, heavily reliant on raw or lightly cooked fish and marine mammals, supplied abundant vitamin D and omega-3 fatty acids, protecting against rickets and cardiovascular dysfunction despite the absence of agricultural produce. In Mesoamerica, the process of nixtamalization, soaking maize in alkaline lime water, released niacin, preventing pellagra, a deficiency disease marked by dermatitis, diarrhea, and dementia. These methods reflect empirical recognition that food preparation itself could transform health outcomes. The line between sustenance and medicine blurred most strikingly in the deliberate selection of specific foods to address particular ailments. Garlic was not only a flavoring agent but a therapeutic tool, its sulfur compounds lowering blood pressure and combating infections. Ancient Egyptian medical papyri from around 1500 BCE list garlic as a remedy for circulatory and pulmonary conditions. Similarly, onions, rich in flavonoids, were employed across Eurasian cultures as treatments for respiratory complaints. These practices anticipated biochemical discoveries millennia later, embedding nutritional pharmacology within everyday life. The role of cereals and legumes extended beyond caloric supply to metabolic regulation. The combination of grains with pulses, as in rice with lentils or maize with beans, provided complete amino acid profiles that supported growth and immune function. Archaeological records from the Indus Valley Civilization and pre-Columbian Americas show such combinations as dietary staples. While the biochemical rationale was unknown, the empirical results, stronger, more resilient populations, ensured the persistence of these pairings. Milk, too, carried both risks and benefits. Lactose intolerance was common among early adult populations, yet fermentation into cheese or yogurt reduced lactose content while preserving proteins, calcium, and fats. Genetic studies reveal that lactase persistence, the ability to digest lactose into adulthood, emerged independently in Europe, Africa, and the Middle East around 5,000 to 7,000 years ago. This mutation conferred nutritional advantages where dairy animals were domesticated, linking diet directly to genetic evolution. The interplay between genetic adaptation and cultural practice reinforced the medical dimension of food, turning milk into a renewable source of nutrition and hydration during periods of scarcity. Food also played a role in modulating inflammation and recovery from injury. Fatty fish, abundant in coastal diets, supplied long-chain polyunsaturated fatty acids with anti-inflammatory properties, reducing swelling and supporting wound repair. Nuts and seeds provided essential minerals such as magnesium and zinc, critical for enzymatic processes in tissue regeneration. These contributions may not have been consciously categorized as medical, but their effects were evident in recovery rates and vitality. The preservation of surplus food likewise had medical implications. Smoking, drying, and salting inhibited bacterial growth, extending shelf life and reducing the likelihood of famine-related malnutrition. Salt, while a preservative, also functioned as a therapeutic agent. Saline washes were used to cleanse wounds, and salt solutions alleviated dehydration long before the development of modern oral rehydration therapies. Archaeological discoveries of salt production sites, such as those in Hallstatt, Austria, dating back to the Bronze Age, underlined centrality to survival. The symbolic dimensions of food reinforced its medicinal role. Ritual feasts often featured items regarded as strengthening or protective, embedding nutritional value within cultural practice. Honey, milk, and specific grains were offered to deities, reflecting both reverence and recognition of their life-sustaining properties. These symbolic associations ensured continuity of beneficial foods across generations, securing not only nourishment but embedded forms of preventive medicine. The recognition of food as a healing force transformed subsistence into a therapeutic act. Every meal carried the possibility of harm or restoration, shaping bodies not only through calories but through chemistry. Yet even with careful diet and preparation, invisible dangers lingered in the environment, pathogens borne by water, soil, and air, that demanded strategies not of ingestion but of avoidance and ritualized control. Human exposure to disease was not determined solely by diet or injury, but also by the conditions of daily life. Water sources, burial practices, waste disposal, and patterns of settlement all mediated the spread of infection. Long before germ theory articulated the microbial origins of disease, cultural behaviors emerged that reduced pathogenic exposure, not through conscious scientific reasoning but through ritual, taboo, and tradition. These practices were adaptive, embedding health protection within symbolic frameworks that preserved them across generations. Water held central significance. Rivers and springs offered hydration, fish, and fertile soils, but they also carried pathogens. Communities that developed rules governing water contact reduced the risk of gastrointestinal illness. Archaeological evidence from the Indus Valley Civilization, flourishing around 2500 BCE, reveals an elaborate system of wells, baths, and drainage channels. While these reflect an advanced level of urban engineering, they also embody a cultural emphasis on ritual purity. Daily bathing and segregation of clean and dirty water sources minimized exposure to contaminated fluids. Comparable practices in ancient Egypt, where Nile water was filtered through porous clay jars, demonstrate convergent adaptations that limited ingestion of harmful microbes even in the absence of microbiological knowledge. Fire provided another dimension of protection. Beyond its role in cooking and warmth, controlled burning acted as a sanitary measure. Ashes were spread over refuse, inhibiting flies and suppressing odors associated with bacterial decomposition. In many societies, ritual purification through smoke, using resins such as frankincense or juniper, was believed to expel malevolent spirits. Modern chemical analysis of these resins has shown antimicrobial activity against airborne pathogens, suggesting that symbolic purification had tangible hygienic benefits. The association of fire with cleansing became deeply embedded in religious practice, from Zoroastrian fire temples to Vedic ritual offerings, ensuring continuity of practices that incidentally reduced disease spread. Burial customs also reveal the intersection of ritual and hygiene. The prompt interment or cremation of the dead limited exposure to decaying bodies, which attract vectors and harbor pathogens. The transition from exposure burials to formalized interments reflects both symbolic reverence and pragmatic disease control. In ancient Greece, edicts required burial outside city walls, reducing contamination of urban centers. Among the Yanomami of South America, endocannibalistic funerary practices involved consuming ashes rather than leaving corpses exposed to insects, inadvertently eliminating a reservoir for pathogen transmission. These cultural responses, though framed in cosmological terms, functioned as early public health measures. Food taboos carried similar protective consequences. Prohibitions against consuming spoiled meat, carrion, or shellfish in certain seasons reduced exposure to toxins and microbial infection. In the Mosaic dietary laws, avoidance of pork and shellfish is framed in religious language, yet epidemiological studies suggest that trichinosis and shellfish poisoning posed genuine risks in the ancient Near East. Whether consciously recognized or not, the codification of such prohibitions embedded protective dietary practices within the authority of divine mandate, ensuring adherence through belief rather than empirical explanation. Domestic environments reveal further layers of ritualized hygiene. In Neolithic settlements such as Çatalhöyük in Anatolia, dating to 7500 BCE, houses were regularly replastered with clay and whitewash containing lime. Lime is strongly alkaline and possesses antimicrobial properties, reducing bacterial survival on surfaces. Archaeologists interpret this as both aesthetic renewal and ritual purification, yet the health benefits would have been substantial. Similarly, the periodic abandonment and rebuilding of dwellings in many early agrarian societies prevented accumulation of waste and parasites, serving as a structural reset against disease vectors. The significance of ritual cleansing extended into bodily practices. Among Hindu populations, the tradition of ablution in rivers, such as the Ganges, carried deep spiritual meaning, yet daily bathing also reduced ectoparasite load and skin infections. Islamic injunctions requiring ablution before prayer institutionalized regular washing of hands, face, and feet. In Jewish tradition, the mikveh ritual bath reinforced periodic immersion. These practices, though religious in rationale, institutionalized behaviors that suppressed microbial transmission long before science identified the mechanism. Taboos extended to reproductive and sexual domains. Restrictions on intercourse during menstruation, quarantine after childbirth, and purification rituals for new mothers served not only symbolic functions but also reduced exposure during vulnerable physiological states. Anthropologists studying traditional societies note that these restrictions often coincided with periods of heightened infection risk, offering inadvertent protection. By embedding such practices within cultural law, compliance was achieved at scale, creating systematic reductions in morbidity. Archaeoparasitological evidence underscores the impact of hygiene practices. Coprolites from prehistoric latrines reveal high burdens of helminth eggs, including whipworm and roundworm, in societies lacking sanitation. Yet comparative data show reduced prevalence in regions with ritualized waste disposal. The Minoan civilization of Crete, for instance, developed terracotta pipes for latrine drainage by the 2nd millennium BCE. While framed as engineering innovation, the reduction of soil contamination with feces curtailed parasitic transmission, improving community health. The persistence of ritual hygiene across cultures reflects its dual role as both a marker of identity and a safeguard against invisible threats. Social enforcement mechanisms, shame, taboo, divine command, ensured compliance in ways that practical reasoning alone may not have achieved. These behaviors demonstrate how symbolic systems acted as carriers of adaptive knowledge, binding survival strategies to cosmology. Yet ritual and hygiene could not eliminate the threat of widespread contagion. Populations continued to confront periodic waves of epidemic disease that no amount of purification or taboo could forestall. When infections spread beyond the capacity of communal practice to contain them, the consequences reshaped societies, altering demography, power structures, and the genetic landscape of entire populations. No matter how carefully communities managed food, hygiene, or ritual, pathogens periodically surged beyond the capacity of cultural practices to contain them. Epidemics punctuated human history with devastating force, leaving demographic scars and reshaping the trajectories of entire societies. Before the existence of formal medicine, populations confronted infectious waves armed only with inherited immunity, environmental adaptations, and the blunt instrument of isolation. These episodes of mass mortality reveal how deeply disease influenced the survival of humanity. Archaeological and textual records document the recurrent emergence of smallpox, plague, cholera, typhus, and other contagions. Variola virus, the causative agent of smallpox, left its characteristic imprint on mummified remains from Egypt, including those dated to the reign of Ramses V in the 12th century BCE. The pustular lesions visible on the skin of preserved bodies confirm that this disease afflicted civilizations thousands of years before inoculation. Genetic reconstructions of ancient viral DNA suggest smallpox diversified as early as the 1st millennium BCE, spreading along trade routes that interconnected Africa, Asia, and Europe. The high mortality among immunologically naive populations periodically reset demographic balances, with survivors passing on genetic or acquired resistance. The Antonine Plague, which struck the Roman Empire beginning in 165 CE, illustrates the transformative potential of epidemics. Contemporary accounts by Galen describe fevers, diarrhea, and pustules consistent with smallpox. Mortality estimates suggest up to 10% of the empire's population may have died over the course of the outbreak. The epidemic altered the balance of military power, strained agricultural output, and undermined economic networks, revealing the capacity of pathogens to destabilize even complex state structures. Such events, while catastrophic, also intensified selective pressures on immunity, favoring alleles that improved viral resistance. Bacterial diseases shaped human populations with equal force. Yersinia pestis, the bacterium responsible for plague, spread in repeated waves across Eurasia. The Justinianic plague of the 6th century CE, described by Procopius, decimated Constantinople and surrounding regions. More than 800 years later, the Black Death of the 14th century annihilated nearly half the population of Europe within a few years. Recent studies of skeletal remains from mass graves in London and Denmark have identified genetic shifts during this period, including changes in immune regulatory genes such as IRAP2. These findings show that epidemics left not only historical but genomic footprints, reshaping the very biology of populations that endured. Cholera exemplifies the role of waterborne transmission in amplifying mortality. Long before Robert Koch identified Vibrio cholerae in 1884, periodic outbreaks devastated South Asia and, through global trade, reached Europe and the Americas. Severe dehydration caused by massive fluid loss could kill within hours. Communities without knowledge of rehydration therapy had few defenses other than fleeing contaminated environments. Genetic evidence shows that blood group O, more prevalent in certain populations, increases vulnerability to severe cholera, shaping the geographic distribution of disease severity. The rise and persistence of cholera demonstrate the fragility of human survival in the face of microbial exploitation of essential resources such as water. Isolation emerged as one of the few effective strategies available to societies without medicine. Archaeological evidence from leprosaria, specialized settlements for individuals with Hansen's disease, demonstrates how communities sought to contain chronic infection by social segregation. Quarantine, later formalized in Venetian ports during the 14th century, originated as an empirical recognition that restricting movement could reduce spread. While often framed in religious or moral language, these practices curtailed transmission long enough for outbreaks to subside, though at enormous human cost to those stigmatized. The psychological impact of epidemics compounded their biological toll. Art, literature, and ritual reflect recurring attempts to make sense of mass mortality. In medieval Europe, flagellant movements interpreted plague as divine punishment, enacting public penance in hopes of appeasing supernatural forces. In other regions, scapegoating led to persecution of minorities accused of spreading disease. These cultural responses highlight the desperation of populations deprived of explanatory frameworks that could identify microbial causes. Yet beneath the symbolism, the brute fact remained. Only those with favorable immune responses or chance avoidance survived. Epidemics also reshaped geopolitics. The arrival of smallpox in the Americas after 1492 annihilated indigenous populations who lacked prior exposure. Mortality estimates exceed 70% in some regions, destabilizing political structures and facilitating European conquest. Entire empires, including the Aztec and Inca, collapsed under the combined weight of warfare and disease. This pattern underscores the asymmetry of immunological history. Exposure over centuries had hardened Old World populations, while isolation had left New World societies acutely vulnerable. The demographic transformation of the Americas cannot be disentangled from the microbial exchange that accompanied global contact. Modern genetic studies continue to reveal the long-term consequences of these ancient crises. The CCR5 delta 3-2 mutation, a deletion in the chemokine receptor gene, confers resistance to HIV infection. Its relatively high prevalence in European populations has been hypothesized to result from historical selective pressure by plague or smallpox, though debate continues. Regardless of precise origin, the mutation exemplifies how survival through epidemics etched adaptations into the genome, shaping the landscape of vulnerability and protection inherited by modern humans. Yet epidemics were never uniform in their outcomes. Some communities, through geography, social organization, or sheer luck, escaped the worst effects. Island populations, remote mountain groups, and nomadic bands sometimes evaded exposure entirely, though their lack of acquired immunity left them fragile in the long term. The unevenness of epidemic impact contributed to shifting balances of power, enabling some societies to expand while others contracted under demographic collapse. Even as populations learned to recognize patterns of contagion, their means of response remained constrained. Ritual purification, food taboos, and isolation offered partial barriers, but the underlying threat persisted. Communities confronted not only sudden waves of mortality, but also the enduring burden of survival with chronic conditions left by disease. And where acute epidemics raged, the violence of injury demanded yet another realm of intervention: surgical attempts to repair bodies under conditions of extraordinary risk. Injuries inflicted by hunting, combat, or accident often demanded more than splinting or herbal application. When bleeding was severe, infection advanced, or bone shattered beyond natural repair, intervention required cutting, scraping, or removal of tissue. Long before the emergence of modern surgical science, humans developed practices that involved direct manipulation of the body with sharp tools. These procedures were conducted without anesthesia, antisepsis, or an understanding of anatomy, yet they achieved survival in cases that otherwise would have been fatal. Trepanation remains the most widely documented prehistoric surgical procedure. Across Europe, Africa, Asia, and the Americas, thousands of skulls dating from the Neolithic onward display circular or rectangular perforations. Survival is confirmed by evidence of bone remodeling around the openings, sometimes extensive enough to indicate decades of life after the operation. Techniques varied: scraping with flint until the inner table was breached, drilling with rotary tools, or cutting with sharp stone blades. The motivations remain debated: ritual release of spirits, treatment of seizures or head trauma, but the frequency and success of trepanation indicate that early surgeons acquired practical skill. They learned to avoid major blood vessels, to halt when the dura mater was reached, and to care for wounds long enough for recovery. Amputation represents another sphere of surgical intervention. The 2022 discovery of a 31,000-year-old skeleton in Borneo with a cleanly amputated leg, healed without fatal infection, demonstrates that complex surgery was performed tens of millennia before agriculture. The precision of the cut and the patient's survival suggest not accidental trauma, but deliberate removal, likely to prevent gangrene after injury. Comparable, though later cases are found in Egyptian and Greek medical texts, which describe amputations for necrotic limbs. Without anesthesia, such procedures depended on physical restraint and crude analgesics: alcohol, opium derivatives, mandrake root, none sufficient to abolish pain but capable of dulling perception. Hemostasis was achieved through methods available in nature: cauterization with heated metal or stone sealed blood vessels and reduced hemorrhage, though at the cost of severe burns. Application of styptic powders containing alum or plant-derived tannins constricted tissue and stemmed bleeding. Tourniquets, described in Indian surgical texts such as the Sushruta Samhita, c. 600 BCE, were used to control circulation during limb operations. These methods illustrate an empirical recognition of vascular control, centuries before Harvey described the circulation of blood in the 17th century. Wound debridement, the removal of damaged or infected tissue, was another early surgical practice. The Edwin Smith papyrus from Egypt, dating to around 1600 BCE but likely copying older sources, describes lacerations treated by excision and bandaging. Honey and grease were applied as dressings, both soothing tissue and reducing microbial load. Surgeons lacked antiseptic theory, but repeated observation taught that some substances improved outcomes. The combination of cutting and dressing represents a rudimentary yet effective surgical protocol. In the context of warfare, surgical necessity intensified. Arrow extractions, documented in Assyrian reliefs, required enlarging the wound tract with knives or probes to remove embedded projectiles. Roman military medicine, as described by Celsus and later Galen, institutionalized surgical care in field hospitals, valetudinaria. Soldiers underwent amputations, excisions, and fracture reductions, all without anesthesia. Survival depended on speed, cleanliness of instruments, and the resilience of the patient's immune system. Instruments recovered from Pompeii and other Roman sites, bronze scalpels, forceps, probes, demonstrate the technical sophistication reached within classical antiquity. Pain management remained the greatest limitation. Herbal sedatives such as opium, cannabis, henbane, and mandrake were administered orally or as inhaled fumes. These induced drowsiness, reduced anxiety, and dulled sensation, but they did not eliminate consciousness. Alcohol, widely available in fermented form, produced intoxication that blunted perception of pain. Mixtures of these agents, sometimes combined with physical restraint, allowed operations to proceed. While crude by modern standards, these methods enabled procedures that saved lives otherwise lost to injury or infection. Cultural interpretations framed surgery within broader systems of meaning. In ancient India, the Sushruta Samhita not only catalogued procedures but emphasized training, recommending practice on gourds and animal carcasses before human application. In ancient China, early texts describe acupuncture combined with minor surgical interventions, embedding bodily manipulation within cosmological theory. These traditions demonstrate that surgery was not seen as purely mechanical, but as part of an integrated worldview, combining empirical technique with philosophical justification. Despite successes, mortality remained high. Without antisepsis, surgical wounds were vulnerable to gangrene and sepsis. Without transfusion, blood loss was often fatal. Yet evidence of survival, healed amputations, remodeled trepanations, scarred but functional limbs, shows that surgery was not mere desperation but an established survival strategy. It required courage of both patient and practitioner, and it demanded communal support during recovery, as immobilized individuals could not survive without provisioning. Surgery without anesthesia illustrates the extremes of human adaptation, the willingness to inflict controlled trauma to prevent greater harm. It reveals both ingenuity and limitation, a capacity to intervene directly in the body without knowledge of its microscopic realities. These practices, though perilous, ensured that individuals with injuries beyond the scope of herbal medicine or ritual healing could sometimes return to life. Yet surgical interventions did not exist in isolation. They were embedded within broader medical systems that combined observation, philosophy, and accumulated practice, forming traditions that endured across centuries and regions. As human societies expanded beyond small foraging bands into complex agrarian and urban civilizations, healing practices became increasingly systematized. What had once been experiential knowledge of herbs, wounds, and rituals was codified into formal traditions that combined empirical observations with philosophical frameworks. These systems, though divergent in theory, converged on the attempt to explain disease within coherent models that could guide treatment across generations. Their endurance demonstrates both the adaptive value of organized medical thought and the capacity of culture to preserve and refine health practices. In South Asia, the development of Ayurveda illustrates the sophistication reached in early medical philosophy. Texts such as the Charaka Samhita and Sushruta Samhita, compiled between the 1st millennium BCE and the early Common Era, outlined comprehensive theories of the body based on the balance of doshas, Vata, Pitta, and Kapha. While framed in metaphysical terms, these treatises included extensive catalogues of herbs, minerals, surgical techniques, and dietary prescriptions. The Sushruta Samhita describes hundreds of surgical instruments, many analogous to modern scalpels, forceps, and probes, and provides detailed instructions for procedures ranging from cataract extraction to rhinoplasty. Empirical efficacy was intertwined with cosmological interpretation, producing a system that addressed both physical symptoms and spiritual balance. In China, the emergence of traditional Chinese medicine, TCM, followed a comparable trajectory. The Huangdi Neijing, Inner Canon of the Yellow Emperor, compiled during the late Warring States and early Han periods, articulated a theory of health grounded in qi, the vital energy flowing through meridians of the body. Illness was attributed to blockages, imbalances, or external perturbations of this flow. Treatments included acupuncture, moxibustion, herbal prescriptions, and practices such as qigong and tai chi that integrated movement with internal regulation. While the theoretical framework differed radically from modern physiology, many TCM remedies contained pharmacologically active compounds later identified in laboratory settings. Artemisinin, derived from Artemisia annua and used for centuries in the treatment of intermittent fevers, became the foundation of modern antimalarial therapy after its isolation by Tu Youyou and colleagues in the 1970s. Egyptian medicine combined practical observation with ritualized prescription. The Ebers Papyrus, dating to around 1550 BCE, contains over 700 remedies, including applications of honey, castor oil, and willow bark. It describes techniques for suturing wounds, setting bones, and reducing swellings, alongside incantations designed to ward off malevolent forces. Egyptian physicians specialized in particular domains, dentistry, ophthalmology, or abdominal complaints, reflecting an early form of professional differentiation. Their practices influenced neighboring civilizations through trade and conquest, diffusing knowledge across the Mediterranean world. The Greco-Roman tradition, crystallized in the works of Hippocrates, Galen, and later Roman compilers, emphasized humoral theory, the balance of blood, phlegm, black bile, and yellow bile. While conceptually distinct from doshas or qi, humoralism represented another attempt to rationalize illness as imbalance rather than supernatural affliction. Greek physicians emphasized observation of symptoms, prognosis, and the role of environment. Galen's anatomical studies, though limited by prohibition against human dissection, systematized medical thought for more than a millennium. His influence stretched from the Roman Empire into the Islamic world, where scholars such as Avicenna synthesized Greco-Roman, Persian, and Indian traditions in works like the Canon of Medicine, which remained a cornerstone of medical education in Europe until the 17th century. These systems were not static but dynamic, evolving through interaction. Trade routes such as the Silk Road facilitated exchange of medicinal plants, instruments, and ideas. Cinnamon, pepper, and other spices traversed continents, valued as both culinary and therapeutic agents. The spread of paper and translation movements in Baghdad's House of Wisdom during the Abbasid period preserved and disseminated texts from diverse traditions, ensuring continuity of medical knowledge across linguistic and cultural boundaries. Despite their metaphysical frameworks, many practices embedded within traditional systems displayed measurable efficacy. Turmeric and ginger, staples of Ayurvedic prescriptions, exhibit anti-inflammatory and digestive benefits confirmed by modern pharmacology. Acupuncture stimulates endorphin release and modulates neural pathways, offering analgesic effects documented in clinical trials. Egyptian use of honey as a wound dressing aligns with its antimicrobial properties. These continuities under the empirical roots of practices that survived because they produced observable results, even when cloaked in symbolic or cosmological language. At the same time, traditional systems carried limitations. Reliance on bloodletting in humoral medicine often weakened rather than strengthened patients. Use of toxic substances such as mercury or arsenic in various traditions caused harm under the guise of therapy. The interpretive dominance of metaphysical frameworks sometimes constrained revision, discouraging departure from established doctrine even when outcomes were poor. Yet the resilience of these systems reflects their broader cultural role. They provided not only treatment but also meaning, embedding illness within narratives that could be understood and managed. The endurance of these traditions also reveals the intersection of medicine with authority. Court physicians served rulers, temples codified rituals of healing, and texts became instruments of professional status. Healing was not only a matter of survival, but also of power, identity, and legitimacy. Those who mastered specialized knowledge gained social influence, ensuring the preservation of practices across centuries. Traditional medical systems represent a stage in human adaptation where practical experimentation converged with philosophical speculation. They preserved and transmitted therapeutic practices, structured training of healers, and created intellectual frameworks that organized human experience of illness. Their influence persisted long after their origins, shaping both regional practices and global medicine. Yet human survival depended not only on abstract systems, but on adaptation to the harsh demands of environment, where extremes of climate, scarcity, and ecology required their own forms of ingenuity. Human adaptability was tested most severely at the margins of habitable landscapes. Arctic tundra, arid deserts, and dense tropical forests imposed physiological and cultural demands that shaped survival strategies. In these contexts, the distinction between subsistence and medicine became blurred, as the very techniques used to endure extreme conditions doubled as protective measures against illness and debilitation. In Arctic regions, populations such as the Inuit developed dietary systems that counteracted deficiencies inevitable in environments with minimal plant growth. Reliance on marine mammals and fish provided not only calories but essential micronutrients. Raw consumption of seal liver and whale skin supplied vitamin C, preventing scurvy in the absence of fruits and vegetables. High intake of omega-3 fatty acids from marine oils contributed anti-inflammatory benefits and supported cardiovascular resilience under cold stress. Anthropological studies of Inuit communities reveal low incidence of cardiovascular disease historically, an outcome linked to these dietary patterns. Fermentation of fish and meat, common in Arctic storage practices, also enriched microbial diversity in the gut, strengthening digestion and immune function. Cold exposure itself demanded physiological adaptation. Shivering thermogenesis generated immediate heat, while long-term acclimatization altered metabolic rates and peripheral blood flow. Culturally, insulated clothing from animal skins, layered with air-trapping furs, provided protection superior to woven fabrics until the advent of industrial textiles. Snow houses, igloos, with their paradoxical warmth due to compacted snow's insulating properties, exemplify environmental engineering that reduced risk of hypothermia and frostbite. In combination, these strategies allowed human survival in latitudes where unprotected exposure would result in rapid death. In desert environments, survival hinged on the conservation and strategic use of water. Populations such as the Bedouin of the Arabian Peninsula developed fluid management practices that minimized dehydration. Camel milk, rich in electrolytes and relatively stable under heat, served as both food and hydration source. Dates, abundant in oasis regions, provided glucose and potassium, buffering against electrolyte loss. Clothing adapted for airflow and sun protection reduced water loss through perspiration while limiting direct exposure to solar radiation. Ritualized avoidance of certain foods or activities during peak heat, often embedded in cultural calendars, reinforced physiological prudence. Medicinal plant use in deserts reflected deep ecological knowledge. Species such as comifera, myrrh, and aloe vera offered antimicrobial resins and soothing gels for skin lesions exacerbated by arid climates. Salt deposits, collected from dry lake beds, were dissolved for wound cleansing and dehydration treatment. Bedouin traditions included preparation of teas from desert herbs with diuretic or antipyretic properties, regulating fluid balance and alleviating fevers. While derived from local flora, these practices mirror pharmacological strategies documented across more temperate societies, adapted here to the scarcity and intensity of desert ecology. Tropical rainforests posed an opposite challenge: abundance of plant life combined with high pathogen load. Constant humidity favored proliferation of fungi, bacteria, and parasites, exposing populations to a spectrum of infections. Indigenous groups developed extensive pharmacopoeias drawn from surrounding biodiversity. The Amazonian use of quinine-containing cinchona bark for malarial fevers represents one of the most famous examples, later foundational for modern anti-malarial therapy. Dozens of rainforest plants contain alkaloids, terpenoids, and glycosides with antimicrobial or analgesic properties, many still under pharmacological investigation. Navigation of toxic flora and fauna demanded acute discrimination. The use of curare, derived from lianas such as Chondrodendron tomentosum, as arrow poison illustrates a sophisticated understanding of dose. While lethal in hunting, its muscle-relaxant properties in controlled amounts were applied to treat convulsions and rigidity. Venoms, both feared and studied, became part of survival strategies. Ethnographic reports describe topical application of diluted snake venoms or insect secretions for analgesic or ritual purposes, reflecting an experimental engagement with biochemical extremes. Housing and settlement patterns further shaped survival in tropical environments. Elevated dwellings reduced exposure to damp ground, snakes, and insect vectors. Smoke from indoor fires, while irritating to lungs, acted as a deterrent against mosquitoes, reducing malaria and yellow fever transmission. Body painting and the application of plant-based oils created physical barriers against biting insects, combining cultural expression with practical protection. In each case, the strategies were embedded in daily life, inseparable from identity yet serving unmistakable health functions. High-altitude environments introduced additional constraints. Populations in the Andes and Himalayas adapted to hypoxia through distinct physiological pathways. Genetic studies of Tibetan populations reveal mutations in the EPAS1 gene, regulating hemoglobin production to optimize oxygen transport without increasing blood viscosity. In the Andes, higher hemoglobin concentrations served as an alternative adaptation. Coca leaf chewing among Andean peoples alleviated fatigue and altitude sickness through stimulation of respiration and vasoconstriction. While initially a cultural ritual, its widespread use demonstrates its role in sustaining productivity under hypoxic stress. The diversity of human strategies in extreme environments underscores the integration of ecological knowledge, cultural practice, and physiological resilience. Each adaptation carried both protective and medical dimensions. Diets countered deficiency, clothing prevented exposure, rituals regulated dangerous behaviors, and plant use modulated disease risk. These practices were not incidental, but central to survival at the margins of habitability. Yet even as human ingenuity allowed survival in deserts, tundras, and jungles, another domain of vulnerability remained universal. The processes of reproduction, childbirth, and infancy carried dangers that no climate or ecological adaptation could fully eliminate. Maternal mortality and infant loss shaped demographic structures, demanding collective strategies and ritual responses that left profound imprints on human history. Reproduction represented both continuity and peril for human communities. The passage from gestation to birth exposed women and infants to dangers that were pervasive across pre-modern societies. Without surgical intervention, antisepsis, or systematic obstetric knowledge, childbirth was one of the leading causes of mortality for women of reproductive age. The survival of infants was equally uncertain, subject to infection, malnutrition, and congenital vulnerabilities. The demographic stability of populations depended on strategies that mitigated these risks through collective practice, ritualized assistance, and the gradual accumulation of empirical techniques. Archaeological evidence demonstrates that maternal mortality was a persistent reality. Skeletal remains often reveal pelvic deformities consistent with obstructed labor, while burials of women alongside neonates testify to deaths during childbirth. In prehistoric cemeteries across Europe, a significant proportion of female deaths cluster in the 20 to 35 age range, corresponding to peak reproductive years. Historical records confirm the magnitude of this danger. In medieval Europe, estimates suggest maternal mortality rates approaching 1 to 1.5% per live birth, translating to a lifetime risk of death exceeding 10% for women bearing multiple children. Comparable rates were observed in early agrarian societies across Asia, Africa, and the Americas. In the absence of formal obstetrics, midwives emerged as crucial figures. Ethnographic parallels among traditional societies highlight the role of experienced women in guiding delivery, recognizing malpresentation, and employing manual techniques to assist passage. Some archaeological contexts provide indirect evidence of midwifery. Small figurines depicting women in squatting or kneeling birthing postures, such as those from the Neolithic Near East, reflect cultural codification of delivery practices. Squatting, by widening the pelvic outlet and utilizing gravity, facilitated birth more effectively than supine positions later imposed by institutional medicine. Herbal remedies supported the process. Plants with oxytocic properties, such as blue cohosh, Collophyllum thalictroides, in North America, or ergot, Claviceps purpurea, in Europe, were administered to stimulate uterine contractions. While dangerous in excess, controlled use could shorten labor and reduce exhaustion. Analgesic herbs, including opium poppy and cannabis, alleviated pain, though their efficacy was limited by dosage variability. Aromatic oils and resins served antiseptic functions when applied to the birth canal or umbilical stump, reducing infection risks before the microbial basis of puerperal fever was known. Ritual dimensions framed childbirth as a liminal state, requiring protective ceremonies to guard against malevolent forces. In Mesopotamia, incantations invoked deities to safeguard mother and infant, while amulets inscribed with protective symbols were placed in birthing chambers. In Mesoamerican traditions, the temazcal, a sweat lodge, was used postpartum for purification and physical recovery. These rituals, though symbolic, often carried practical benefits. Isolation of the birthing woman reduced contamination, and postnatal seclusion allowed maternal rest and neonatal bonding. Infant mortality remained high despite these practices. Osteological analyses reveal evidence of malnutrition, rickets, and infectious disease in skeletal remains of children. Estimates for prehistoric and early historic populations suggest that as many as 30 to 50% of children died before reaching the age of 5. Breastfeeding provided some protection, delivering maternal antibodies through colostrum and extending nutritional support in environments where alternative feeding carried contamination risks. Weaning, however, was fraught with danger. Transition to soft foods exposed infants to waterborne pathogens, increasing rates of diarrheal mortality. In some societies, prolonged breastfeeding into the second or third year reduced this risk, though it imposed energetic demands on mothers. Communities employed diverse strategies to buffer infant survival. Wet nursing spread the burden of lactation, while cross-suckling among related women distributed risk. Ritual protection extended to neonates, who were often swaddled in amulets, painted with symbolic pigments, or shielded by charms placed near cradles. These practices reinforced social investment in fragile lives, ensuring vigilance during the most perilous years. Burial customs for infants, frequently distinct from those of adults, highlight the recognition of their vulnerability and the communal weight placed upon their survival. Demographic resilience depended on high fertility to offset losses. Ethnographic and historical data indicate that women in pre-industrial societies commonly bore 6 to 8 children, of whom perhaps half reached adulthood. This reproductive intensity was not merely cultural but biological necessity in maintaining population stability. Periods of famine or epidemic magnified infant mortality, leading to temporary population decline. Yet recovery was possible through sustained fertility, supported by social norms encouraging early marriage and multiple pregnancies. The psychological toll of high infant mortality is difficult to reconstruct, but ethnographic observations suggest a dual dynamic of grief and acceptance. Naming practices often delayed formal recognition of infants until survival past early months, reflecting uncertainty. At the same time, elaborate mourning rituals for deceased children indicate the depth of attachment when survival was cut short. These cultural mechanisms both acknowledged the reality of loss and reinforced the community's capacity to persist. The biology of reproduction also imposed constraints on maternal survival. Pelvic morphology, shaped by bipedal locomotion, created a narrower birth canal than in other primates. Combined with large neonatal head size, this resulted in frequent obstructed labor. Human birth was consequently a social event, rarely solitary, necessitating assistance for both safety and success. This dependency reinforced cooperative structures within communities, embedding reproductive survival into collective responsibility. Advances in archaeological genetics provide additional insight. Mutations such as those affecting vitamin D metabolism, critical for skeletal development, may have influenced rates of obstructed labor by shaping pelvic morphology. Selection pressures from maternal mortality interacted with broader survival dynamics, etching reproductive hazards into the evolutionary record. Survival of both mothers and children hinged not only on biological luck, but on the cumulative strategies of kinship, ritual, and ecological adaptation. Even when childbirth succeeded, the infant remained precarious, dependent on immune protection from breast milk and the vigilance of caregivers. The fragility of these early years left indelible marks on demographic structure, shaping lifespans, family organization, and the rhythm of human continuity. Yet alongside these vulnerabilities, deeper genetic defenses were accumulating, biological adaptations forged in the crucible of disease, written into the genome as enduring records of survival and loss. The human genome carries the imprint of ancient struggles with pathogens. Every epidemic and every endemic disease exerted selective pressure, favoring those individuals whose genetic variants conferred even marginal advantages in survival. Over millennia, this process accumulated into a complex archive of resistance traits, many of which remain visible in modern populations. The study of these adaptations provides insight not only into past mortality, but also into the enduring biological legacy of humanity's battle with disease. One of the most thoroughly studied examples is the sickle cell trait. The point mutation in the HBB gene substitutes valine for glutamic acid in the beta-globin chain of hemoglobin. Homozygous carriers develop sickle cell disease, characterized by distorted red blood cells and systemic complications. Yet heterozygous individuals gain resistance to severe Plasmodium falciparum malaria. This balanced polymorphism, maintained at high frequencies in sub-Saharan Africa, parts of the Middle East, and India, illustrates the paradox of genetic survival. A mutation lethal in some contexts is preserved because it protects against a more common and deadly threat. Molecular studies confirm that parasitized red blood cells carrying sickle hemoglobin are eliminated more efficiently by the spleen, reducing the likelihood of overwhelming infection. Other malaria-related adaptations demonstrate the multiplicity of evolutionary responses. The absence of the Duffy antigen receptor, DRC, widespread in West African populations, prevents entry of Plasmodium vivax into red blood cells. Glucose 6-phosphate dehydrogenase, G6PD deficiency, while predisposing to hemolysis under oxidative stress, offers partial protection against malaria by increasing the vulnerability of infected erythrocytes. These traits, scattered across regions of high malarial burden, underscore the intimate relationship between vector-borne disease and human genetics. Beyond malaria, resistance adaptations emerged from repeated encounters with viral epidemics. The CCR5 delta 3-2 mutation, a 32 base pair deletion in the CCR5 gene, renders the receptor non-functional, blocking HIV entry into host cells. Its high prevalence in European populations, estimated at up to 10% in some regions, suggests historical selective pressure predating the HIV pandemic. Hypotheses link its survival advantage to resistance against smallpox or plague, though debate continues. Ancient DNA studies indicate the mutation was present in Northern Europe more than 5,000 years ago, supporting the idea that earlier pathogens maintained its frequency. Lactase persistence offers another striking example of adaptation, though its relevance extends beyond nutrition into disease resistance. In populations that adopted dairy pastoralism, the mutation allowing continued lactase production into adulthood provided access to a safe, renewable source of hydration and calories. In environments where contaminated water carried pathogens, milk consumption reduced exposure to waterborne diseases. The independent emergence of lactase persistence mutations in Europe, Africa, and the Middle East demonstrates convergent evolution shaped by the interplay of diet, environment, and microbial threat. The evolution of immune system genes reflects constant selective tension. Human leukocyte antigen, HLA, loci display extraordinary diversity, with hundreds of alleles maintained in global populations. This polymorphism enhances the capacity of immune systems to recognize a wide array of pathogens. Genomic comparisons with Neanderthals and Denisovans reveal that interbreeding contributed additional immune-related alleles, including variants in toll-like receptors that improved recognition of bacterial and fungal components. These introgressed genes likely enhanced survival as Homo sapiens expanded into Eurasia, encountering novel pathogen ecologies. Autoimmune disorders, paradoxically, may represent the cost of heightened immune sensitivity. Alleles conferring strong pathogen resistance also predisposed to immune dysregulation in environments with reduced microbial burden. For example, HLA variants associated with protection against leprosy and tuberculosis are also linked to susceptibility for type 1 diabetes and celiac disease. This trade-off highlights the dynamic equilibrium between survival advantage and pathological consequence, a balance shifted by changing environments. Genetic resistance also influenced population-level outcomes during historical epidemics. The Black Death in 14th-century Europe, caused by Yersinia pestis, reduced populations by as much as half in some regions. Recent analyses of skeletal DNA suggest selective survival of individuals carrying variants in immune regulatory genes such as IRAP2, which enhances pathogen recognition. These alleles remain at higher frequencies in modern descendants, evidence of rapid selection under conditions of extreme mortality. Even structural genetic changes can reflect ancient disease pressure. Thalassemias, widespread in Mediterranean and South Asian populations, reduce hemoglobin production, leading to anemia in homozygotes but protection against malaria in heterozygotes. The persistence of these disorders in high frequency illustrates the intensity of selective forces exerted by infectious disease, maintaining deleterious alleles because of their protective benefits in heterozygous carriers. The study of ancient DNA has expanded understanding of these dynamics. Sequencing of remains from Bronze Age Eurasia reveals that many immune-related alleles rose in frequency during periods of dense urbanization and increased trade, when pathogens spread more efficiently through large populations. Cities became crucibles of selection, where only those with adequate genetic defenses survived repeated outbreaks. The very existence of modern populations reflects centuries of filtration through waves of mortality. Resistance adaptations extend beyond individual genes to entire physiological systems. Variability in innate immune receptors, cytokine production, and fever responses reflects evolutionary fine-tuning under pathogen pressure. The capacity for fever, while metabolically costly, enhances immune efficiency and accelerates recovery from infection. Genes regulating inflammatory pathways demonstrate balancing selection, maintaining responsiveness without tipping into excessive damage. These subtle calibrations shaped survival at both individual and collective scales. The accumulation of resistance traits, however, did not provide invulnerability. Pathogens themselves evolved in response, generating new strains capable of bypassing defenses. Influenza, with its high mutation rate, evaded immune memory repeatedly, ensuring recurrent outbreaks. Tuberculosis, adapting to host immune pressures, persisted as a chronic scourge. The human genome recorded victories in the form of protective alleles, yet the struggle remained ongoing, an evolutionary arms race without resolution. These genetic legacies, visible in contemporary variation, reflect the cumulative survival of countless ancestors who endured epidemic after epidemic. Yet they also illustrate constraints, embedding vulnerabilities and predispositions into populations. Every adaptation came at a cost, shaping physiology in ways both protective and pathological. As humans inherited these imprints, their capacity to endure disease became not only a biological matter, but also a foundation for the future of medicine itself, where ancient strategies and modern science would converge. The knowledge embodied in human survival without medicine is not confined to the past. It informs the trajectory of contemporary biomedical science, shaping research agendas and guiding public health strategies. The practices and adaptations forged through millennia of necessity provide both cautionary examples and reservoirs of insight, reminding us that modern medicine rests on a foundation laid by countless generations who endured without it. One lesson lies in the pharmacological potential of natural products. Many of the most effective drugs in current use trace their origins to plant, fungal, or microbial compounds first encountered through empirical healing traditions. Aspirin derives from salicylates in willow bark, morphine from the opium poppy, quinine and later artemisinin from anti-malarial plants. Even antibiotics, the hallmark of 20th century medicine, originated from soil organisms whose antimicrobial activity was observed and cultivated. The continued search for bioactive molecules in biodiversity-rich environments reflects this lineage, as pharmaceutical exploration returns repeatedly to nature for templates of therapeutic innovation. Equally instructive is the recognition that immunity is not static. The genetic traces of past epidemics illustrate the dynamic balance between human defenses and microbial evolution. Pathogens exploit ecological and social conditions, and selective pressures continue to shape responses. The COVID-19 pandemic has underscored the vulnerability of global populations to emergent viruses, echoing historical experiences of immunologically naive communities confronting novel infections. Understanding ancient selective events provides a framework for anticipating future risks, informing strategies for vaccine development and genomic surveillance. The role of cooperation emerges as another enduring lesson. Archaeological evidence of survival among disabled or chronically ill individuals highlights the evolutionary advantage of caregiving. In modern societies, this principle extends into public health, where collective measures, vaccination, sanitation, epidemiological monitoring, determine outcomes as decisively as individual resilience. The persistence of social responses to disease, from ritual quarantine to contemporary lockdowns, demonstrates continuity in the logic of containment. While the tools have changed, the underlying principle, that survival depends on shared responsibility, remains constant. Food, once a medium of survival and medicine, continues to shape health outcomes. Nutritional epidemiology confirms the protective effects of diets rich in diverse plant compounds, mirroring ancient practices that combine sustenance with therapy. The microbiome, now recognized as a determinant of immunity, reflects dietary histories that once depended on fermentation, fiber, and microbial exposure. Lessons from traditional diets emphasize the dangers of monotony and the importance of biochemical diversity, insights that resonate amid rising chronic disease linked to industrialized food systems. Traditional medical systems, though shaped by cosmologies distinct from modern science, also offer perspective. They remind us that medicine has always been embedded in cultural meaning, not merely in technical intervention. Healing involves both physiological repair and symbolic reassurance, as evident in the placebo effect and in the psychological benefits of trust in practitioners. Modern medicine, with its emphasis on randomized trials and molecular mechanisms, risks overlooking this dimension. Incorporating lessons from traditional systems does not mean adopting their metaphysics, but recognizing that the human experience of illness requires both biological and existential care. The adaptability displayed in extreme environments offers further guidance. Inuit survival on marine diets, Andean acclimatization to hypoxia, Bedouin water management, and Amazonian ethnobotany exemplify human ingenuity in tailoring strategies to ecology. As climate change alters global environments, these precedents highlight the capacity of cultural and biological flexibility to mitigate new stresses. They also remind us that resilience requires integration of ecological knowledge with survival practice, an insight increasingly urgent in a world of shifting disease vectors and resource scarcity. Modern genetics extends these lessons into the laboratory. The discovery of resistance alleles, such as CCR5 delta 3-2 or sickle cell trait, underscores how ancient survival strategies inform contemporary therapies. Gene editing technologies, such as CRISPR-Cas9, now explore ways to replicate or harness these adaptations in treating diseases ranging from HIV to malaria. Understanding the evolutionary costs of such traits, autoimmunity, anemia, metabolic compromise, provides caution, emphasizing that interventions must account for trade-offs inscribed by natural selection. The continuity between ancient and modern survival strategies is perhaps most visible in vaccines. Inoculation and variolation, practiced in China, India, and the Ottoman Empire before their adoption in Europe, represent the culmination of centuries of empirical engagement with immunity. These practices embody the principle that exposure, managed in controlled form, can protect against catastrophic disease. Vaccination today extends this logic with molecular precision, yet its conceptual roots lie in pre-modern recognition that survival could be engineered through deliberate intervention. The lessons are not merely historical, but prospective. They invite reflection on what future descendants may infer from the medical practices of the present. Just as we scrutinize trypan skulls and papyri for evidence of adaptation, future societies may evaluate antibiotics, vaccines, and gene therapies as provisional tools in a continuing struggle. The evolutionary arms race with pathogens persists, and the strategies of today will one day be seen as steps in a much longer continuum of survival. The endurance of humanity without modern medicine demonstrates that resilience emerges from a combination of biology, culture, and environment. It underscores that adaptation is not solely technological, but also genetic, dietary, social, and symbolic. Yet the very same forces that preserved life in the past now intersect with unprecedented challenges: urban density, ecological disruption, microbial resistance, that extend the struggle into new domains, demanding strategies as inventive and multifaceted as those of our ancestors.